Hi, my name is Juan Rendón, and my presentation's title is Influence of Female Size on the Frequency of Multiple Paternity in the Bronze Mudhorn Shark Mostellus henley. Polyandry is one of the most common shark mating systems. Polyandry also tends to be coercive on behalf of males, which has led to intersexual conflicts and antagonistic coevolution. In general, females seek to avoid damage to themselves during mating, while males seek to counter this behavior. Male biological features have developed to promote polyandry and can include hooks or spores on their claspers to increase the chances of fertilizing female ova and sperm that are suited to the conditions of the female reproductive system. Males also exhibit a biting behavior during mating. In response to this, females have developed thickened skins to protect themselves. Females also show a sheltering behavior and sexual segregation to avoid mating, in addition to cryptic postcopulatory selective processes that occur within their umbilical glands. Mostellus henley, also known as the bronze mudhorn shark, is a small placental viviparous species that is distributed from the California coast to Ecuador and Peru, including the Gulf of California. Mostellus henley can be distinguished from other species of the same genus by the presence of frayed edges on the dorsal and caudal fins, and also by the particular shape of its teeth. Previous studies have demonstrated that reproductive isolation among populations of this species occurs in the northeastern Pacific. This reproductive isolation can promote variation in some parameters, such as the minimum reproductive size, litter sizes, and frequencies of multiple paternity. Gestation period, however, remains similar among populations with only small variation. Given that several studies have evaluated possible differences in both maternal size and litter size between monandrous and polyandrous females, and that a positive linear relationship between maternal size and litter size has been found for Mostellus henley in the northern Gulf of California, we established the following research questions. First, we ask, what is the frequency of multiple paternity in the upper Gulf of California? We also ask, is the frequency of multiple paternity greater among larger females? To answer these questions, we evaluated multiple paternity and its relationship between maternal size and litter size in Mostellus henley from the northern Gulf of California. We genotyped 4 microsatellite loci in 23 grade females and 113 embryos. We then evaluated the number of sires for each litter and then identified the contribution of each sire to each litter. We found that of the 23 litters analyzed, 13 were sired by multiple males. The frequency of multiple paternity was more than 50%, which is a relatively intermediate value when compared to the records of multiple paternity for this species in the northeastern Pacific. This frequency of multiple paternity is also intermediate when compared to those of other species that show seasonal monogamy, such as Squalus mitsukuri, Squalus acanthias, and Styria nativura, and species with high frequency of polyandry, such as Negaprium brevirostris, Carcharias taurus, and Carcharinus blombeus. We evaluated ship relationships using the colony software. The software assigned each of the embryos to either groups of full seedlings or half seedlings based on their genotypes, maternal genotypes, and paternal genotypes that were inferred using the GERUD program. On both the X and Y axis, we have the different IDs for all the embryos in this study, and the yellow, blue, purple, and orange diamonds up here represent full ship relationships, while the green triangles here represent half ship relationships. We found evidence of skewed male reproductive success, which was more apparent in the largest litters. In general, a single dominant male cited more than 40% of the embryos in all litters. This suggests the presence of postcopulatory sexual selection at work. In particular, this skew in male reproductive success may be attributed to cryptic selection acting on the sperm from multiple males. This process likely occurs in the umbilical gland of the female. We used a generalized linear model to assess the relationship between multiple paternity with both maternal length and litter size. Given that maternal length and litter size were considered redundant variables, only maternal length was included in the model as the independent variable, with the number of sires as the response variable. Since we found a maximum of 4 sires in this study, 4 scenarios were established, the ones with the highest probability of occurrence being located at the extremes of the graph. On the x-axis, we have the different maternal lengths that ranged from 61.6 centimeters to 88 centimeters. And on the y-axis, we have the different probabilities of occurrence for each of the 4 scenarios. The curves represent each scenario. We found that females with total lengths between 60 and 65 centimeters were more likely to have higher probabilities of their eggs being fertilized by a single male, while females with total lengths between 90 and 95 centimeters would be more likely to have higher probabilities of their eggs being fertilized by 4 males. We also carried out a Mann-Whitney test in order to assess possible differences in both maternal length and litter size between polyandrous and monandrous females. We found significant differences in both of these variables. If we extrapolate the results of the generalized linear model to the other 2 populations of Mustelus henley in the northeastern Pacific, we may predict that females from California will have a higher probability of their eggs being fertilized by a single male, while females from Baja California Sur will have a higher probability of their eggs being fertilized by multiple males. In conclusion, Mustelus henley from the northern Gulf of California presented a slightly greater frequency of multiple paternity than that of the California population, but about half the frequency of the Baja California Sur population. The paternal reproductive skew suggests that postcopulatory cryptic selective processes are at work. There were significant differences in both maternal length and litter size between monogamous and polyandrous females, and latitudinal variation in the reproductive parameters in the 3 Mustelus henley populations was present. Finally, I would like to thank to all the people and institutions involved in the development of this project.